So now we're going to be discussing decision-making framework for microbiome sequencing. So the step one that we start with is defining the research goal. Who is there? Firstly, taxonomic census only. So use targeted amplicon sequencing, 16S, 18S, or ITS. Best for broad surveys of microbial diversity, hundreds or thousands of samples epidemiological scale, low biomass samples, biopsies, urine, hot springs. Limitation is that we have low resolution, often genus level, not strain level, cannot profile viruses, plasmids, or functional genes. Secondly, who is there and what they can do. So functional profiling. First we did taxonomic profiling, then we do functional profiling, who is there and what they can do. So use whole genome shotgun WGS with Illumina. This is best for studies requiring gene pathway analysis, metabolism, antibiotic resistance, virulence. Then moderate number of samples where functional resolution is critical, detecting bacteria, fungi, viruses in one dataset. Limitation is that more expensive than Amplicon, around 5 to 10 times more expensive than Amplicon, computationally heavy, and many may fragment assemblies, short reads. Next up, we have high resolution genome assembly. We can use long read WGS, that is PacBio Hi-Fi, or ONT, that is Pacific Biosciences or Oxford Nanopore Technologies. So this is best for novel genome discoveries using MAGs, plasmids, phages, etc. Resolving repeats and structural variants, strain level tracking and outbreaks, for example, resistant E. coli strains, and epigenetic analysis. PacBio for methylation, ONT for 5-MC6-MA detection. Limitation for this is higher cost, more demanding DNA extraction, requires high molecular weight DNA, and historically higher error rates, though now Hi-Fi and duplex solve this. Then we consider the sample type, right? Whether we have soil or environment, human gut, low biomass, virus focus study, pathogen outbreak tracking, right? So we consider the sample type. For soil or environmental, we use the method Amplicon for diversity surveys, WGS for function and novel enzymes. Rationale behind that is high diversity, so we need balance of cost and resolution. That is why we use Amplicon. Then human gut or stool, Amplicon large scale cohort surveys to WGS for functional gut profiling, cost versus function trade off. Low biomasses, biopsies, urogenital skin, again Amplicon because it is sensitive and works with low DNA yield. Moving on, virus focus study, we use WGS, whole genome sequencing, using Illumina or ONT, no universal viral marker genes. Then pathogen outbreak tracking, long read WGS like Pacific Biosciences or ONT, which is, again, I'm repeating, Oxford Nanopore Technologies, because strain level differentiation is essential here. And single cell specialized profiling, again, PacBio, Hi-Fi, or ONT, since we can resolve single genomes with long contiguity. Then we check for the budget and resources. If we have low budget and we have many samples to cover, we use Amplicon. If moderate budget and functional focus, Illumina WGS. And if high budget and high resolution focus, PacBio or ONT WGS. Now, what's the main research question? We check only community census if we have to do Amplicon. Then taxonomy and functional analysis, Illumina WGS. Strain resolution, novel genomes, epigenetics, PacBio, ONT. How many samples? Hundreds, thousands if we have, then Amplicon, which is cost effective. If we have tens of hundreds, Illumina WGS is best for that. If we have few but we want to deep dive, we have to go in depth, then we use PacBio or ONT. And sample constraints, low DNA yield given by Amplicon, viral fungal inclusion by WGS, and epigenetics needed, then we use PacBio or ONT. So that's about our first basically summary. Let us summarize. Amplicon for cheap census, Illumina WGS gives a functional snapshot, and PacBio ONT WGS genome level and epigenetic detail. Right. Now moving on. So basically now we have to talk about judging the possession-specific sequence quality from chromatogram to FastQ. So in Sanger sequencing, results are seen as chromatogram. Colored peaks represent bases A, T, C, or G. The height and sharpness of peaks denotes the signal strength, and the overlapping or the noisy peaks denotes lower confidence. Now, modern NGS doesn't produce chromatograms for each read, but the core idea remains the same. That is, each base call has an associated confidence score. Now, FastQ format. So each read in the FastQ format has 4 lines. Firstly, we have the header, that has the read ID, and the instrument information. Then we have the sequence, that is the actual called DNA basis, A, T, C, or G. Then the separator line denoted by plus, starting with plus. Then the quality string, that is ASCII character encoding the Fred quality scores Q, right? So for example, we have the FastQ line in this FastQ format line in this format, ASCII ID, sequence ID. Then the basis, the sequence, the actual DNA basis, A, G, C, T, A, C, D, G, right? And then we have plus, that is the separator line. And then we have the ASCII character encoding using the Fred quality score. Now, Fred quality score is basically a logarithmic score expressing base calling confidence, which has the formula Q is equal to minus 10 times log of P error, which is the probability of error, to the base 10. P error is probability that the base is wrong. Now, Q score, and we can check for the error probability. If we have Q10, that means 1 in 10 probability of it being wrong. So the accuracy is 90%. This means this is 10% wrong, right? And Q20 is 20% wrong. Sorry, not 20%, 1% wrong. This is 10% wrong. This is 1% wrong. That is why we have accuracy 99%. Q30 is 0.1% wrong. That is accuracy is 99.9%. And Q40 is 1 in 10,000. That is 0.01% wrong or error probability, giving us 99.9% accuracy. Right. So that is the basic formula. And ASCII encoding, each Q score is represented as a character in the FastQ file. Example, this is what the question mark. It is represented as Q30 and basically has 0.1% error rate. This way, FASTQ compresses thousands or millions of quality values into simple character strings. Importance is that quality scores are essential for filtering, trimming, and error modeling in all downstream analysis. Then we have the adapter, primer, and barcode removal. So why? During sequencing, library preparation and extra sequences are added. Adapters allow the DNA fragments to bind the flow cell or nanopores. Allows DNA fragments to bind the flow cell. Then primers initiate the sequencing reactions, and barcodes are indices which distinguish samples in multiplex runs. If they are not removed, adapters primers appear in reads, contaminating the downstream analysis and can cause incorrect alignments, chimeras, or false taxonomic hits. Solution is we use pre-processing tools like Trimomatic, CutAdapt, and FastP to remove adapters or primers, and QIMA2 and DALA2 pipelines also filter low quality ends and truncate reads based on the Q scores. Now, multiplexing and demultiplexing, the problem that we have is single sample sequencing. It generates way more data than one sample needs, and it is very costly per sample. Therefore, we have a solution that is multiplexing. That is, we pool multiple samples together in one sequencing run, add a unique barcode or an index sequence to each sample's fragments during the library preparation, and the sequencer reads the insert biological DNA and the barcode, which is the sample ID. Now, demultiplexing is after sequencing, the software reads back into the sample specific bins by looking at the barcodes. For example, sample 1 fragments belongs to barcode 1, sample 2 for barcode 2. After sequencing, all reads are split back correctly. Result is that each sample gets enough data and overall run becomes cost efficient. Putting it all together, the pipeline, so sequencing machine output, we have FASTQ files with sequences plus the quality scores. Quality control, we just using the per base quality using Fred scores which have been assigned ASCII values. Trim or remove low quality reads. Then we have the adapter primer removal, clean reads from technical sequences, perform demultiplexing, that is assign reads back to samples using barcodes, and downstream analysis like alignment, taxonomic classification, variant calling, functional profiling, right? So that's about it. And in summary, FredQ scores basically contain the confidence in base calls that are crucial for quality filtering. Adapter primer removal is necessary to avoid technical artifacts, and multiplexing, demultiplexing makes sequencing economical by pooling samples with barcodes.